Hello, good afternoon. Today I am present my reading report on the soil organic nitrogen dynamic during the grassland restoration from a plant AMF microbe perspective. The central message is that the restoration should not only be evaluated by the visible vegetation recovery but also whether nitrogen is retained stabilized below grounds. This presentation focus on one central question: how the plant AMF and the soil microbe work together in the rebuild the stable soil organic nitrogen during the grassland. I will first introduce why soil organic nitrogen is important, then explain biological mechanism, evidence, and future direction. This framework organizes the literature into four connected layers. Environmental stress initiates degradation, while the plant AMF and microbes regulation nitrogen transformation and retention. The final goal is not only vegetation recovery but the formation of stable soil organic nitrogen pools. This slide presents the central mechanism of my report. During the grassland restoration, plant provide the organic input that support the soil nitrogen cycling. AMF connect the plant roots with the soil environment and enhance the nutrient exchange. Microorganisms transform and retain nitrogen through biomass formation and residue production. When these products are protected by soil aggregate and minerals, stable soil organic nitrogen pools are developed. Vegetation recovery is important, but it does not always indicate that the function have recovered. Soil organic nitrogen integrate plant input, microbial transformation, AMF activity, and soil protection process. Therefore, the soil organic nitrogen provides a better indicator for a long-term restoration. The previous published results are showing that the plant do not respond only to total nitrogen availability. They use different nitrogen form, and these uptake patterns reflect the soil nitrogen cycling and influence below ground restorations. Uh, Another study: during the restoration, plants and microorganisms compete for available nitrogen. All thought the microbial immobilization may temporarily reduce the plant nitrogen availability. It retains nitrogen within the soil system. After microbial turnover, these retained nutrients can contribute to stable organic nitrogen formation. AMF functions beyond the nutrient sequestration. The hyposphere creates a special microbial environment where the organic nitrogen transformation, microbial immobilization, and nitrogen retention are connected through these interactions. AMF can strengthen the internal nitrogen cycling and reduce the potential nitrogen losses. The functional changes can appear before the visible biomass structure. The nitrogen uptake pattern and plant microbial competition can provide early indicator of ecosystem recovery and nitrogen limitations. AMF functions beyond the nutrient sequestration through the hypha and microbial interaction and the mineral protection. AMF contributes to organic matter stabilization and support the long-term soil organic nitrogen recovery. AMF diversity and glomaline formation can improve the soil aggregation and protect organic matter. The formation of POC and MAOC pools support the long-term carbon and nitrogen retention. Nitrogen addition can reorganize microbial network, but more nitrogen does not always improve the restoration. Ecosystem recovery depends on microbial functions and nutrients balance. Long-term restoration depends on microbial network stability, evoluting, and microbial functions together with vegetation recovery provide a better understanding of ecosystem resilience. The degraded grasslands may be more sensitive to excessive nitrogen inputs. Microbial stability should be considered when designing restoration strategies. The future studies should combine isotope tracing, soil fractionation, and microbial residue analysis and AMF function indicator to directly test nitrogen stabilization mechanisms. These slides highlight the main research gap. Most restoration studies use plant biomass, soil organic carbon, or short-term nitrogen uptake as indicator, but these do not directly prove stable soil organic nitrogen recovery. Future work should combine long-term restoration gradients with 15 and nitrogen isotope tracing to directly test AMF-mediated nitrogen transfer and stabilization into PON nitrogen and MAON nitrogen fractions. These are the future research direction gap. Here is the literature which I have used in my book report also and in this presentation. Thank you so much.